Cardiac muscle contracts through excitation of cardiac myocytes. As just mentioned, the specialized cells capable of generating an electrical impulse are found only at nodal sites in the heart, the SAN and AVN. Since the SAN normally discharges at a higher frequency than any other region, it acts as the pacemaker for the entire heart. From here, the electrical impulse travels across the atria to the AVN, from where when calcium enters the cardiac muscle cells during the plateau phase of the action potential and stimulates the release of calcium from the cardiac muscle sarcoplasmic reticulum, the muscle contracts. There are a few differences between the contraction of skeletal and cardiac muscle. Cardiac muscle action potential has a plateau due to the influx of calcium ions. Cardiac nodal cells spontaneously depolarize, resulting in automaticity. And cardiac myocytes are electrically coupled to each other by gap junctions. It is important to note that the action potential of cells from different regions of the heart are not The contraction of cardiac cells is similar to other excitable cells and depends upon permeability to and movement of sodium, potassium, and calcium ions. The contractile cycle can be divided into phases, each of which has an ionic basis. Phase 0 The action potential that was just described is typically seen in atrial and ventricular myocytes and Purkinje fibers. Remember, phase 0 is the rapid upstroke as a result of the voltage-gated sodium channels opening. Phase 1 is the initial repolarization when the voltage-gated sodium channels are inactivated and the voltage-gated potassium channels begin to open. Phase 2 is the plateau phase, whereby the calcium influx through voltage-gated calcium channels balances potassium efflux. The calcium influx triggers calcium release from the sarcoplasmic reticulum, which triggers myocyte contraction. Phase 3 is rapid repolarization as a result of a massive potassium efflux due to opening of voltage-gated slow potassium channels and closure of voltage-gated calcium channels. And finally, phase 4 is the resting potential. The pacemaker action potential is seen in sinoatrial nodes and atrioventricular nodes. It differs from the ventricular action potential in the following ways. First, phase 0 is not as rapid since these cells do not have voltage-gated sodium channels. Second, the phase 2 plateau is not seen in pacemaker cells. And third, the phase 4 depolarization is slower than that. The influx of calcium during the cardiac action potential is the key to making myocytes contract, which is the formation of the heartbeat. It is calcium binding to muscle-specific proteins that drives the consumption of ATP that is used during muscle cell contraction. Relaxation of cardiac myocytes requires removal of free calcium from within the cell. As just discussed, it is the generation of electrical current through the movement of ions across the cell membrane that drives influx of calcium into the cell, which is why the process is called excitation-contraction coupling, with calcium being the link between the excitation and the contraction. An electrocardiogram, or ECG, demonstrates the changes in membrane potential that occur during the cardiac cycle. In a typical ECG plot, we will find a P wave, which represents atrial depolarization, the PR interval, which shows the conduction delay through the AV node, the QRS complex, which shows ventricular repolarization, the QT interval, which demonstrates the mechanical contraction of the ventricles. The T wave shows ventricular repolarization. The ST segment is isoelectric when the ventricles are depolarized, and a U wave is seen in disorders such as hypokalemia and bradycardia. The cardiac cycle is described as one complete sequence of contraction and relaxation and can be divided into 5 phases. Phase 1 is isovolumetric contraction, the period between mitral valve closure and aortic valve opening during which the highest oxygen consumption occurs. Phase 2, systolic ejection, is the period between the aortic valve opening and closing. Phase 3, isovolumetric relaxation, occurs between the time when the aortic valve closes and the mitral valve opens. Phase 4 is the rapid filling period just after the mitral valve opening, and Phase 5 occurs just before the mitral valve closes and is known as reduced filling. As the valves open and close, and the chambers of the heart empty and fill, there are distinct sounds that can be heard. In a complete cardiac cycle, these are S1 from mitral and tricuspid valve closure, S2 from aortic and pulmonary valve closure, S3 in beginning in 2008, the USMLE Step 1 exam included audio questions such as heart sounds. Be sure you are very familiar with the sounds we just reviewed, as they are likely to show up on the Step 1 exam. Now let's take a look at the coronary arteries. The right and left coronary arteries arise from the right and left sinuses respectively. In most cases, the SAN and AVN are supplied by the right coronary artery. In a right dominant system, the right coronary artery supplies the inferior portion of the left ventricle via the posterior descending artery. The left anterior descending artery that supplies the anterior interventricular septum is where coronary artery occlusion most commonly occurs. It is the coronary arteries that fill up during diastole. The most posterior part of the heart is the left atrium. Enlargement can cause dysphagia or hoarseness. Now let's look at a few commonly used formulas used to evaluate cardiac function. Cardiac output can be obtained by multiplying stroke volume times heart rate. Since the stroke volume initially goes up during exercise, the cardiac output also increases. With longer duration of exercise, the heart rate also goes up and the cardiac output increases even more. However, if heart rate is too high, diastolic filling is incomplete, and the cardiac output decreases. This is typical of conditions Now let's take a look at preload and afterload. The preload is the ventricular EDV and the afterload is the mean arterial pressure. The preload increases with exercise, increased blood volume, and excitement. It is the preload that pumps up the heart. Venodilators act to reduce the preload, whereas vasodilators act to reduce the afterload. The intrinsic relationship between the end diastolic volume, or EDV, and the stroke volume is known as the Frank-Starling law of the heart. A plot of the stroke volume against the preload is known as the Starling curve. It demonstrates that the force of contraction is proportional to the initial length of cardiac muscle fiber, or preload.